That's not happening. Yeah, no, that's not going to, he's not writing a chapter.
Yeah, so I don't know if that changes part of his decision.
No, it's possible he'll meet somebody there who wants to work with him on a chapter, but that's not how it typically works. Oh, we're ready to go. Can you hear me in the back? Are we okay? Sound? So it looks like we have about a third of the class actually comes to lectures. I think I'm going to cancel having online lectures. What do you think? No. It's boring. No one comes to class. Why am I here? I could be on a beach in Hawaii broadcasting. It's a waste of my time, to tell you the truth. But anyway, with all that said, I'll decide over spring break what I'm going to do. But don't be surprised if there's no more online course feeds. Okay, anyway, with that grim beginning to the talk, kind of sad Stephen Hawking with ALS died. Brilliant guy. He had this indolent form of ALS, which can go on for a long time. Most people with ALS, unfortunately, die within 2 to 5 or 6 years. But a brilliant guy. It shows you what happens. You can have severe disability and still be just a brilliant contributor to the human race. So let's finish the part of the class on brain tumors. We talked about primary brain tumors that grow inside your cranial vault, metastatic brain tumors that start somewhere else in your body and end up in your brain. This is incredibly common occurrence. Metastatic tumors being the highest form of brain tumors. But then when they present, they can have swelling. So the patient can look really sick because they get cerebral edema. And what do I mean by that? What I mean by that is this is a normal cell here. And around the cell, this is a blood vessel, a little capillary in your brain. And around it, you have a kind of, the capillary endothelial cells. And then you have these little astrocytes around it. And what happens with tumor-related edema, this is called vasogenic edema. And it's called vasogenic edema because what happens is the connections between the lining cells of your capillaries here, these tight junctions, the tumor secretes a toxic factor that opens up these junctions. And what happens is that protein spills out of your blood into the substance of the brain, and that attracts water, and so you get swelling or edema. It's no different than if you were punched in the shoulder and you got swelling, right? There'd be increased fluid in the tissue. You got a knock on the head, a knot on your head. Most of that knot's not blood. It's actually water or edema. So this is vasogenic edema. In tumor, here's an example. Here's a big tumor deep in the brain, and you see all this white stuff here, all that. That's vasogenic edema. It's not the ventricle. There's the ventricle here. And the ventricle, of course, is enlarged because the tumor's blocking cerebral spinal fluid flow, but all of this here is cerebral edema. It's increased water. And you can see it actually has the same density as the ventricle. So, again, edema. So that's tumor-related edema. It's pretty easy to treat. You just give the person corticosteroids, which many of you have had. You might have had a rash and you put steroid on an itch or something. You give it systemically, and it actually, in about 24 hours, fixes this leak in the tight junctions here. Actually, I got a better idea. I got a good idea. Maybe I'll develop some code to the people in the class that if I do some kind of specific movement on a piece of the slide without talking, you will know that it will be on the exam. How about that? It's a solution. Okay, let's try it. Okay, good. Did you get it, guys? That's the solution. I just realized nonverbal communication because the laser pointer is not taped. How great is that? Just came up with that. Yeah. Uh-huh. Sure. That's why I did it, but this is the result. 1/3 of the people come to class, the rest are having cappuccino. It's fine. I don't care. I got a solution. I already came up with the solution. No, this is about normal for the class. I've been here for the whole. Thank you. You know, I'm all for walkouts. I mean, I got arrested for anti-war demonstrations. I mean, I've got a. I don't know if you saw. I actually have my mug shot, right, with the little, pretty good. Chicago, anti-war. Actually, I applied for global traveler on campus, this thing that you get in and out of the country, and I go there, and I went with my wife. She's from Italy. She's all nervous. I'm not so nervous. I don't care. I go in, and there's 2 people being evaluated, and they ask one person to leave. These are the customs agents. I said, this is going to be interesting. They said, have you ever been arrested? I said, yeah, I was arrested in Chicago for anti-war demonstrations. And they said, well, you were arrested for felony mob action, and they never released the charges, so you have a felony charge against you. It's a riot. I said, great. I said, I'm happy. Good for you. They were like, the customs agents go, what's wrong with this guy? I'm going, well, whatever. Okay, good. I got a plan. I've got a plan. Now I feel good. All right. So that's it. And we give the steroids, and we fix that leak. So then there's another kind of edema, which is really bad because it's not fixable. And that's called cytoxic edema. Cytoxic edema occurs when you have loss of your nutrients, oxygen and glucose, and your cells start to die. And what happens is the cells lose their ability to maintain their membrane pump, and they take in fluid, and the cells swell. So cytoxic edema. So the endothelial cells swell. They're dying. The neurons swell. They're dying. The astrocytes, the glial cells, they swell. They're dying. This is cytoxic edema. There's no real treatment for it other than to quickly try to restore oxygen and glucose levels, but oftentimes it's too late. And this edema is not treatable. And here would be an example. You can see here's your brain, and you can see your normal gyri and sulci, and then you can see this swelling here and this darkening and the loss of the boundaries. This is cytoxic edema from an acute stroke. Now, this is not a bad one. There's not a lot of, it's not good to have it. It's not a massive amount of edema that's going to potentially be fatal. So now what's the problem with having this brain swelling? And this is for any of you who go into anything in the medical field, this is a huge medical emergency. What happens is there's a mass. It could be, for instance, an intracerebral hematoma from hypertension, or it could be a metastatic lesion with lots of edema around it. What happens is there's pressure, and pressure pushes on the inside part of the temporal lobe, the uncus, and that uncus pushes down and presses on the midbrain. And the midbrain, if you remember, that's where you have all the things related to maintenance of consciousness, controlling breathing, etc., and you start to rapidly deteriorate. So you really have to be quick. This is called uncal herniation, basically. It's very common. So here's what happens. Here's somebody here who had a massive stroke. That's a middle cerebral stroke. You can see there's anterior cerebral. There's posterior cerebral here, in here, and you can see this big, swollen necrotic mass. That's someone who occluded a middle cerebral artery. Could be any age, doesn't matter. Could be a young person with an arrhythmia who throws an embolus from the heart. Could be more commonly an older person with atherosclerotic disease. And it's really hard to control. What happens is the brain swells. You can see it actually pushing on your brainstem. See here, here's your substantia nigra, which we've already talked about is lost in Parkinson's disease. Okay, and that you can see the hemorrhage. And this person, you're pressing on the brainstem, your reticular activating system is right there, and the person lapses and goes into a coma. And if you don't do something quickly, they're going to die for sure. You can also see this from problems in the back of the brain. This is cerebellar herniation. So remember, your cerebellum's right here. What you're trying to do is keep your brainstem working so that you're awake, right? You're not going to lapse into a coma. You get a hemorrhage in your cerebellum. This is a typical location for a hypertensive hemorrhage. Big hemorrhage, it presses forward on the brainstem. Again, the person goes into a coma. So these are really true medical emergencies. This particular one, the cerebellar hematomas, if you pick them up pretty quickly, they do very well. The surgeon goes in, takes out the clot, and the person really does, usually has a complete recovery. They'll be left maybe with some cerebellar signs, some, poor coordination, some ataxia, and so on. So let's just do a couple more tumors because we talked, and I want you to just be aware of them. Your pituitary gland sits right there. And you guys know, all of you who've taken biological psychology, et cetera, pituitary gland, hormone regulation, growth hormone, thyroid hormone, sex hormones, et cetera. But it also is susceptible to forming tumors. And I'll just give you a case. A neighbor came over to me, asked me their kid was in the 5th percentile at age 10 for height and weight. And she was complaining of headaches. That means something's wrong with her growth hormone secretion. To cut to the chase, the young girl sent her to UCSF. They have a tremendous pituitary adenoma program. I mean, they're unbelievable. And basically, she had a large pituitary adenoma. The surgeons actually take this out by going through the nose. They do a transsphenoidal approach. They come up through the nose and take it out. She had a total cure. She's actually now, the reason I thought of her is I just saw someone recently, I don't live on that block anymore, and said that the girl's now in college doing well. Okay? So pituitary adenoma, another kind of extra brain. It's between the brain, here's the brain, growing out of the pituitary, pushing up on the brain. So in younger people, it usually presents with hormone deficiencies. But in older people, it often just presents as a big mass that eventually starts pushing on your optic nerves and produces visual changes and all kinds of other miseries. Okay, so that's it. I think you had a pretty good overview of what a brain tumor is, and there are many kinds. And the main thing is to diagnose them and get them fixed as soon as you can. So we've now finished the clinical part of the course. So you guys really should be prepared. You kind of understand the exam. You understand the tools you can use to measure human behavior. And you've seen some of the major disease groups. So now we're going to go into the more fun part of the course in some ways, which is actually trying to understand how brain systems are organized for some of our most core human behaviors. Memory, language, decision-making, emotional regulation, visual perception, and so on. So that'll be the last, maybe it's about 30 to 40, maybe 40% of the course. And it should be good. I hope. All right, so here we go. So first, our own Tolman Hall. Whoops, get over here, you monster. Tolman Hall. Edward Tolman, pretty brilliant guy. Actually, since we're talking about protests, you guys may or may not know that, but back in the 50s, people know about the McCarthy era? Joseph McCarthy? Okay, tell us about Joseph McCarthy. What did that bad boy do? Yeah, well, you said you know about it.
I've heard about it.
Okay, so what? What do you think?
Like the Red Scare, right?
Yeah.
Yeah.
Right. Yeah, he was saying everybody everybody's anybody who's against their views is a communist. So they were trying to, accuse. And if you wanted to be in a union, you're a communist. If you want to have Social Security, you're a communist. And what they enforced, the governor of California actually said that every faculty member, that would be me, had to sign a loyalty oath to the United States of America and to the state of California. Now, that is ridiculous, right? I mean, I'm a U.S. citizen. I don't need to sign any additional loyalty oath to know that I'm loyal to my country. So 300 faculty protested. The administration, the deans, the administrators sided with Reagan, said sign. Reagan fired, I'm not sure it's 200 or 300 faculty. Like me, I would have been fired for sure, because there's no way that I would have signed it. And Edward Tolman led the faculty protest. He was the chair of psychology. And it went to the federal courts. It was like a 2 or 3 year case. And eventually the Supreme Court came back and said it was an illegal firing, and everybody was rehired. And I think it changed the tenor of the campus a little bit. The faculty began to be a little suspicious about administrators in terms of telling people how to live their lives and make political decisions. So anyway, Tolman was great in that sense, but he also was a brilliant theoretician, did some amazing work in spatial memory. And here's a quote from him: "Memory is a gift in nature, the ability of living organisms to retain and to utilize acquired information or knowledge. Owners of biological memory systems are capable of behaving more appropriately at a later time because of their experiences." He also posited that there was a spatial memory system in your brain. Okay, what I want you to do, close your eyes, point to where the exit is. Perfect, okay? You guys know where it is. You have a spatial map of this room. He proposed there was a spatial map based on very simple experiments in rodents. I have another solution. Draw things on the board. I'm learning slowly. I'm going to figure this out. Okay. What he did, he had rodents. And let's say the food source is here. Here's food. Okay? The rodents would be put here, the rat would be put here, and there'd be a maze that they'd have to figure out how to get through to get the food. And the rats figure out that maze, right? They eventually learn, if I go through that track, I'm going to get the food. Okay, simple. Now he does a very simple, elegant second experiment. You take the same rat, this guy right here, you put him in the center of a field with choices like this. He could go in that line, that line, that line, that line, that line, that line, or that line. Remember, this is where the food is. If there's no spatial memory system, the rat doesn't know what arm to take. They have to explore, just like in the maze, he might have to go down this arm, might have to go down this arm, and then keep trying. But guess what? You put the rat in this maze, and they figure out immediately that they take this arm and go right to the food. This was the idea that there's in your brain, just like everybody just showed me by pointing to these exits, you have a spatial organization of the world. 2 years ago, the Mosers, Edvard and Maybert Moser, got the Nobel Prize in Medicine for working on the actual single-cell physiology of spatial memory in the entorhinal cortex of rodents, which, by the way, if you remember, is the area that gets pathological involvement first in Alzheimer's disease. In fact, there was some nice talks about that yesterday at UC Irvine. So that's Tolman, a really powerful figure in many ways, not just scientifically, but also socially. So the big thing in memory to remember, the simple big first fact, is that you have both retrograde memory, that's what you did this morning, what you did last week, what you did in high school, what you did in whatever, in the past. It's past experience. And then you have anterograde memory. That's what you're learning. That's what you're picking up as you go through life. New learning. And anterograde memory can be split up into two principal systems that support it. One is short-term or working memory. That's holding something in your mind's eye. So let's just do a simple, remember when I was showing a neuro exam, I'd say to the patient, remember these 3 words, and then I wait 10 seconds and have the person repeat it. So let's do it. You ready? Baseball, say it. Tree, window. Okay, so now we'll do something else for 10 seconds. I'm checking the patient's reflexes. I'm checking out. What I want to see is, can that person hold that information in a short-term cortical memory store? Let's see. What are the words? Outstanding. So she has intact short-term or working memory. I know she's paying attention. I know she could hold it in her cortex. Now, the next question is, does she store it? You don't want to store those 3 words. I hope not, because they're completely irrelevant. But if you wanted to store them, you have to transfer it from short-term memory, dependent on your cortex, and in large part your frontal lobe, in other areas, and you have to transfer it to long-term memory, something that tomorrow you would remember that you learned baseball, tree, and window, and that long-term storage is the part that's absolutely critically dependent on the hippocampus. And we'll talk quite a bit more about that now and figure out exactly how that works. And actually, when I say figure out how it works, I don't think I'm going to tell you how it works, because the last Society for Neuroscience has about 2,000 to 3,000 abstracts every year on this, and no one's quite totally figured it out. Yeah. Prefrontal cortex, PFC, sorry. Okay, so retrograde, anterograde, and the classic case is the case does that work, you think? Okay, good. The classic case of Scoville and Milner in 1957, published in the Journal of Neurology, Neurosurgery, and Psychiatry, where they had a patient who had severe epilepsy, recurrent complex partial seizures, multiple seizures a day, and he'd had one surgical attempt to treat it by taking out the hippocampus on one side. He still seized, and they went back in and took out the hippocampus. There's the amygdala and the hippocampus. Here's the normal brain. Amygdala, hippocampus. They surgically resected the amygdala and the hippocampus on both sides. Up until that point, no one knew that if you did that to a human being, you would lose all your ability to encode new information. This was the classic case. This was the equivalent in the memory world of Broca's case, or the equivalent of the social emotional world of Phineas Gage's case. So really a seminal observation. And it's kind of crazy, but Brenda Milner is still active. She still teaches, and she's 96, and she's up in Canada. It's kind of amazing. But in any event, this was the classic case showing this, that the hippocampus was critically involved in laying down new long-term memories. And it's been the focus of a massive amount of research since then. Now, patient HM, as he's called, those are his initials. He died about 4 years ago, still with no ability to learn anything new, unless it was something that was not a fact or a piece of knowledge or something, something that might go on in this class or something experienced in the world. And you'll see at the end of this lecture, which won't finish today, that he could learn implicit skills. So if he never rode a bicycle, he could learn how to ride a bicycle. He just wouldn't remember learning that he learned to ride the bicycle. He would have a problem with learning facts and new knowledge. Okay? Patient HM, here we go. Unable to learn any new information after surgery other than implicit things like motor performance, able to remember all memories before his surgery. It's a little bit of an overstatement. There's usually a graded retrograde amnesia so that you're not perfectly clear. Let's say you had a hypoxic event from a cardiac arrest, as you'll see, as a cause of memory problems. Person has damage, knocks out part of their hippocampus. It's not like they're perfect right up to the moment of the cardiac arrest. There's a graded, so the week before may not be so clear. But when they get back a month or 2 months prior to the injury, things will be clear. So it's a graded retrograde amnesia. Intact short-term memory, PFC, there's intact prefrontal cortex. Able to learn new procedural skills. He could learn to ride a bike if he never rode a bike, but he wouldn't remember learning to ride the bike. Pretty amazing. No change in his overall intelligence, language, or perceptual abilities. He was an average IQ person. No change in his personality, although most of these patients with hippocampal damage are kind of flat. And they basically don't, because of the fact that they're not really detecting or responding to new things in the environment. They're absent new learning. They tend to be kind of dull when you actually talk to them. HM is cared for all the time by his mother, who accompanied him where he goes. It happened in 1966. His mother was in Hartford. She was having an operation. He was going to go to Boston. He was tested in Boston for years at MIT, years and years and years. It was his father who packed his clothes for him and brought him to meet Dr. Scoville prior to the journey. The father had taken the patient to visit his mother in the hospital. That very morning, the third such visit that week, by patient HM to see his mother. Yet when we questioned him, he seemed not to remember any of the visits, although he expressed a vague idea that something might have happened to his mother. The patients often have a funny sense of familiarity, right? They know something might have happened, but the facts and knowledge of the event are not there. Yeah. This is after surgery. No. The corpus callosotomies used to be a treatment. And we'll talk about that later in the course when we're talking about disconnection syndromes. Corpus callosotomy, it was basically used for someone who had a focal onset seizure, and then it would spread across the corpus callosum and they'd get a generalized seizure. And this would, the corpus callosotomy stopped the spread of the seizure and restricted it to just a simple partial seizure and not a generalized seizure. We don't do many of them now. I mean, I think in the last 2 years, maybe 3 years, we've had one case that we've had to do a callosotomy on. So it's really, it was a big procedure prior to the advent of other more tailored surgical approaches and better anti-epileptic medications. And as you remember, Professor Chauvel kind of laying out, trying to really localize the source. So still available, but for a very, very small number of people. Yes? Well, actually, that's the classic question. What it tells you, if you can remember old things, your old memories are not stored in your hippocampus. That's the first thing it tells you. And the party line now, and I think it's probably true, and we're going to get into it in a little bit, if you take things in that you're going to remember, the hippocampal system does its magic, and then the actual storage of the information is being widely distributed in the cortex. So you might remember, if you're going to remember this, our interaction, some of the verbal part would be in auditory language area stored. They're looking at me, what might be in visual, it would be, accessed more broadly because you don't, and it can't be in one spot of the cortex because you get a lot of cortical lesions, you don't have severe memory loss. It's more distributed. But it's not in the hippocampus. You're exactly right. It can't be, right? They don't have a hippocampus. So, okay. On a journey to Boston, he kept saying he felt a little uneasy and wondered if something might be wrong with one of his parents, this vague familiarity. Seems like it was my mother, but then that's what I'm unsure about. If there's something wrong with my mother, then it could be my father. He's confused about it. He was uneasy. Something's wrong with one of his parents, but he could never figure out what it was. Gradually, although he was told repeatedly that he could telephone home anytime he wished, he no longer knew why he should do so. It completely disappeared from his thought processes. The next day, he appeared completely unaware that there had been any question of illness in his family. When he asked again who had packed his suitcase, he said, "It must have been my mother. She always does these things." Right? So he kind of filled in and confabulated, even though his mother had nothing to do with packing a suitcase because she was in a hospital. This is the classic case of patient HM. So, per your question, short-term memory and long-term memory must be supported by distinct neural systems, right? Because a hippocampal patient, if I asked them to remember a baseball tree and window, and if they kept rehearsing it, they could remember it till the cows come home. They could just keep it in their cortex, Memories are not stored in the hippocampus. Per your question, they must be distributed throughout the cortex. So in 1937, Pabst, who we're going to talk about in terms of the neurobiology of emotion later in the course, laid out this theory of emotional regulation. And I don't want to get into the details of it now, particularly since he was completely wrong. But the system that, well, almost completely wrong. He did have one part right, the cingulate. He said that this, based on studies of patients with rabies, and we'll talk about this later in the course, is that this was your neurobiology of emotional regulation. What he really laid out is the neurobiology of memory encoding. This is the key. He didn't know it, but that's what he was laying out. Your hippocampus, information comes out of your hippocampus, flows through the fornix into your mammillary bodies, up to your anterior thalamic nucleus, up to the cingulate, back to the hippocampus, and whoosh, back out to the cortex for long-term storage. And we're going to break this down by diseases that affect every part of this long-term memory encoding circuitry. So again, to be clear, we've got the hippocampus to the mammillary body, to the anterior nucleus of the thalamus, to the cingulate and back. The magic happens here. What that magic is, I don't think people really know completely, but there's a lot of work, and there was some elegant stuff that was presented yesterday at the UC Irvine Epilepsy Symposium. Okay, so here's a nice picture. I like this picture from David Amaral, who runs the Mind Center up at Davis, who's done a lot of work in this area. Here's your hippocampus. Very nice structure up around to the mammillary bodies. And then in front of that hippocampus is another very important structure called the amygdala. And as you'll see in a few minutes, the amygdala is like your sensory organ for extraction of emotional information. Your auditory cortex is perceiving sound, your visual cortex vision, your somatosensory cortex tactile. The amygdala is really key for extracting emotional salience from the environment. And I'll show you what I mean by that. Okay, now, back to your question. This is the key anatomical slide that laid out what is likely going on. As you hear me or see me or, well, no one's touching me, but anyway, as you do these things, different areas of the brain are activated. This is an upside-down macaque brain. So here's auditory areas. Here's frontal lobe. Here's visual cortex. The entire cortex has a massive afferent connections to the entorhinal cortex right here. Everything you hear, see, think about, decide you want to store has to come from the cortex into the entorhinal cortex, where it goes into the hippocampus, and I'm going to show you the pathway. The magic happens in this circuitry I just showed you. It comes back to entorhinal cortex, and then there's efferent connections back to the cortex for storage. So we know there's an anatomical infrastructure, right? We know the anatomy. What we don't know is the detailed molecular biology and computational things that go into this storage. But we know quite a bit about the anatomy. This is from Van Hosen, and this figures all the way from 1975, but it still holds quite nicely. It's conserved across species, this system. That's why there's so much research on memory that's done in rodents. So on the left here is a primate. This is a monkey again. In and out from the cortex to the parahippocampal, hippocampal regions, and back out to the cortex. That's you, me, monkeys, baboons, and the same system, the same general system you see in rodents. The cortex is not so big, but into the hippocampus and parahippocampal regions and back on out to the cortex. This is work by Howard Eichenbaum, who's unfortunately died this year, but is a brilliant memory researcher at Boston University. This is a conserved evolutionary system, and this, of course, provides an opportunity to use rodent models to study actually human memory. Right? So there's a lot of that you see if you go to the Society for Neuroscience. Most of the articles or the presentations are not on human neurobiology and memory. They're actually rodent, because in the rodent, those of you who are in cell molecular biology, you can manipulate this receptor, you can do optogenetics, do all these beautiful things to try to tease out exactly what's going on. So let's take a little bit closer look. Let's drill down into what is the neurobiology of memory and what diseases affect it. So the first thing we're going to do is, this is an extraction. Here's the hippocampal mammillary body system. I've just taken it out of the brain. Here it is right here. This is an illustration from the brilliant Netter series. For those of you who go into anything medical, you'll see these books. He's an unbelievable medical illustrator. So here's the hippocampus, and now we're going to do a cross-section. We're going to cut right through the hippocampus, right there, and there it is right here. And we're going to look at this cross-section, and you're going to see this beautiful kind of swirled structure sitting right there. That's your hippocampus proper. Okay. So, how does the world get into the hippocampus to do its magic? I already showed you the gross anatomy. The gross anatomy brings all the cortical inputs down into the entorhinal cortex, And then the entorhinal cortex has a massive input pathway called the perforant pathway. How's that working? Is it working? Am I getting there? Got a new plan? Okay, good. The perforant pathway brings information in from the world into the hippocampus, and it synapses on the dentate gyrus. This is a critical, you see these two interlocking C-shaped structures. The dentate gyrus is critical because the dentate gyrus is also the part in your brains that has heavy neurogenesis. What does that mean? As you sit there, in the next 24 hours, your dentate gyrus is going to produce about 200,000 new cells. Think about that, 200,000. And these are going to be used to fill in and keep your memory structures strong and facilitate your memory capacity. So you come in to the dentate gyrus. The dentate gyrus, then here's the perforant pathway. So you're coming in from the world here, perforant pathway, entorhinal cortex, perforant pathway, into the dentate gyrus. The dentate gyrus now projects what are called mossy fibers into the CA3 region of the hippocampus. And here's the critical synapse right here. The CA3 neurons in your brain have a massive input into the CA1 cells. And it turns out this is a glutamate synapse, right? Schaefer collateral. So glutamate, you say, why is glutamate important? Glutamate is your most common excitatory neurotransmitter. About 80% of your neurotransmitters are actually glutamate dependent. And they regulate all kinds of cell activity. The problem is, if you get too massive a release of glutamate, the glutamate, think of your cell here, and you get this much glutamate, pop good, whatever. Now you get a massive amount of glutamate. The cell starts going, it fires like crazy. It can't support its membrane pump, and it starts leaking, and water starts going in, almost like cerebral edema, and eventually your cell gets killed. This is, if you see somebody who's had a cardiac arrest, this is the part you're trying to salvage, because as the cardiac arrest happens and you drop your glucose, your oxygen to your brain, you have a massive release of glutamate, and the glutamate actually lands on the CA1 cells where it normally goes, but there's too much of it. It fires like crazy. You kill cells. And all you have to do is kill these cells. And the behavioral syndrome in the patient looks the same as if Scoville and Milner took the entire hippocampus out. Looks exactly the same. And this is very important. If you think about why do we want to get why you should all be CPR certified, right? Because you only have about 6, 7, 8, maybe 10 minutes, unless it's a kid and they've been under ice, cold in a lake. You only have at the max 10 minutes to restore proper blood flow, or you're going to end up with damage in a patient who may survive but now has profound anterograde amnesia. They lose the ability to encode new information. Okay? Very important. Very common. Has anybody known anybody who's had a cardiac arrest in the room? Good. Nobody. It happens. My friend's back there. Oh, no, the ER doc's not there. No, he's not here today. We have a couple auditors up the class, and one's an emergency room doc. Kind of nice. Okay, good. All right, so, and here's what these things look like. This is your normal hippocampus, you and I. There's your dentate gyrus, and here's the CA3 cells, and here's the CA1 cells. They look really quite nice. Here's someone with severe hypoxia. The dentate's intact, the CA3's intact. You notice the massive cell loss here? There should be cells all through here. They've all been killed by glutamate toxicity. The same thing can happen with hypoglycemia. There's your dentate cells. Here's your CA1 cells. Notice the cells are gone. And when those cells are damaged, you've cut the encoding network, and the outcome is going to be a patient who has a profound problem in new learning, which is really not a good thing, obviously. Okay, now, What other diseases will do this? So that's hypoxia. We're going to go through the diseases. The next disease that does that, as we discussed in class, is Alzheimer's disease, which typically presents with memory problems. Right? That's the most typical presentation. And the reason for that is if you go back to this basic anatomy here, where everything that you're hearing, and I'm telling you to remember baseball tree and window, if you really want to remember it long term, it's got to go to entorhinal cortex. Turns out that's where the pathology starts in Alzheimer's disease. So actually, to some degree, Alzheimer's is a disconnection syndrome. You've cut the connection of the world that you perceive from going into your memory encoding circuit. Right? So very, very common. And how do we know it starts in entorhinal cortex? Post-mortem studies of people dying with Alzheimer's, early onset Alzheimer's, maybe from a different issue, a heart attack, and actually looking at the cell pathology and seeing the initial depositions of amyloid and tau in this particular area. So that'll do it. Eventually, the Alzheimer's pathology will start chewing up the hippocampus itself, but it really starts in the entorhinal cortex. How about a vascular cause of memory? Remember, your hippocampus has to get blood, right? Where's the blood come from? Well, if we go back to the stroke part of the course, if you remember, there's your big old middle cerebral artery, does about 80% of your entire cortical blood supply. But you also have your posterior cerebral artery in yellow coming off. There's your brain stem off the basilar artery, and here it is. And what you can see is the posterior cerebral artery actually provides blood to the hippocampus. So you can actually get a stroke in your posterior cerebral artery and have a memory problem. And interestingly, most people don't realize this, but your hippocampi are lateralized. Most people think the hippocampus are equal on each side. It's not true. If you're right-handed, your verbal memory is dependent on your left hippocampus. Your spatial memory, as I talked about with Tolman's seminal work, is really distributed in both hippocampi. So a right hippocampal infarct in a patient, they may have trouble rerouting how they got into the lecture hall. If this was the first time they came, they may not remember the spatial tracking that someone brought them into this room and sat them down with. But their verbal memory would be intact. So selective stroke can damage your hippocampus, and here's what it looks like. The posterior cerebral artery, this big artery, actually has a branch off it. And it was described in 1928 by Uchimura. It's called the artery of Uchimura, and it actually provides, off the posterior cerebral, it provides blood supply to the hippocampus. And if you infarct it, here's an acute infarction. This was one of my patients. And unfortunately, he was having heart problems. He actually, unfortunately, died from a heart condition. But he had a stroke in his hippocampus. This is what it looks like chronically. If he had survived on a MRI scan, you can see that the hippocampus is just taken out by a stroke. And at post-mortem, you can see what it looks like. The entire medial temporal lobe is out. Your hippocampus has been infarcted. And the output, for those with good eyes in the room, see your mammillary bodies, boom, boom. Remember I said the hippocampus goes to the mammillary body? If you note, the mammillary body on the side of the hippocampal infarction is actually shrunk. It shrunk because all the output of the hippocampus is gone. Right? So that's an infarction causing this. But another interesting syndrome happens, a transient ischemic attack. We talked about transient ischemic attacks before. Let me just try one more. So this is going to be fun for the rest of the course. Did you get the code? Good. Outstanding. So, I may be losing my mind. Track me. Can you track me for the rest of the course, guys? How's the GSIs doing? Am I losing it or am I okay? You're not sure? You're not positive? Okay. Transient ischemic attack is a warning you're going to get a stroke. We talked about that. If a grandpa at Thanksgiving dinner, his right hand drops and he can't speak for 10 minutes, you better get him into the hospital because he's have a warning he's going to have a stroke in his left hemisphere, and you need to do something to stop it. You can get the same syndrome with a TIA here, and it's a fascinating syndrome. Vertebral artery provides blood supply to both hippocampi. I've already told you that. You get a syndrome called transient global amnesia. This is a really interesting syndrome where, like the patient who has transient ischemia to the left hemisphere has a language problem. The person with transient ischemia to the hippocampal formation, they're awake, they're alert, they can't encode any new information, and they know it, and they're upset about it, right? So that you see the patient, and they talk to you, they're not having a seizure, they're not confused, they just know they can't remember anything. And it will last sometimes 2 hours, sometimes 10 hours, sometimes even more. Transient global amnesia. Acute onset of anterograde amnesia, no retrograde memory loss. They knew what they did the day before. They knew what they did in the morning. Last 2 to 12 hours, the patient can't encode any new information. They can still function. They'll walk and talk, and when you see them acutely, they know there's a problem. And they, doc, what's going on? What's going on? What's going on is they're having transient decreased blood flow to their hippocampal regions from a vascular cause. Oftentimes, it can be precipitated under stress. It's usually transient. I've seen maybe 5 or 6 cases over the year that had the syndrome as a variant of an acute migraine episode. Most migraines are just unpleasant, pulsing, maybe visual changes, but some actually have more complicated prodromes, such as transient aphasia, such as transient global amnesia. But it's uncommon, but, the problem with this whole thing about it's uncommon, if you're the one person in the room that has it, it's the most common thing in the world to you. But you've got to be a little bit careful about statistics when you're dealing with individual human beings. You have to evaluate the person really individually. Usually transient, and it's not benign. If you have a lot of these, this is a study we did years ago, when we looked at patients who, quote, recovered. You do formal testing on them, neuropsych testing, they look normal. But they tell you, doc, my memory's just not as sharp as it was, even though formally they look pretty good on pen and paper. And in fact, when you check these patients and look at their MRI scan, we did 15 patients, every one of the patients has a very small cavitary lesion. See these little teeny lesions here, here, here? They're not supposed to be there. These are little cavitary areas of damage in their CA1 region. They haven't completely knocked it out. So they're still able to function in the world, but they tell you, my memory is just not as clear as it should be. Okay, so we'll stop here. Just to be clear, the exam, we'll finish memory on Monday, but the exam only goes up to today. Okay, so you don't have to, we'll have a study guide out pretty soon. Right now comes the study guide. Don't forget the code. This is on. Everything up till today is on the midterm. Monday is not. Okay? Thank you. Everything I give a code to, you put on the exam.
Yeah, I got a list of them.
I'm done.
You realize,
I'm done.
you realize that
I
when
don't
you
care.
say, let's